Ms. Kulkarni here. In this video, let's continue with medicinal chemistry. Now, we learned in the earlier videos that a medicine can be effective, but at the same point, it can lead to toxic level. So, we need to be always aware of both effectiveness as well as safety. So, let's quickly review what do we mean by toxicity. The unacceptable adverse effect of any drug is called as toxicity. And then there are terms like LD50, ED50, and also TD50. LD50 is the concentration of the drug which will kill 50% of animals in the test sample. ED50 is the concentration of the drug which will bring about the desirable positive effect in 50% of the test sample. And toxic dose TD50 is the amount of drug which will cause toxicity among 50% of the patient. Now again, just to remind you, lower the LD50 value, the more toxic is the drug. And also lower the ED50, that means the drug is more effective. And there is therapeutic index which we calculate by simple ratio of LD50 to ED50 in animals and TD50 to ED50 in humans. Greater the therapeutic index, the safer is the drug. Let's try to understand this concept a little bit more. So let's compare two drugs, one with therapeutic index of 200 and another one with therapeutic index of 2. Which one will be more dangerous and which one will be safer? So for the drug with TI value equal to 200, in order for that drug to be toxic, we need to change the concentration 200 times the effective dosage, which is going to be a rare occasion. So that means this is going to be relatively a safe drug. What about TI equals 2? If we happen to double the concentration of that drug than the effective dose, then that reaches the value of toxic dose. So the medicine becomes extremely toxic. So this will not be a safe drug to use. So how can we correlate therapeutic window with safety? Or what is the perfect dosage for any drug? Look at the graph here. In this graph, we see here the effective dosage and this is the toxic dosage. So the value here is given for the effective dosage. The value here is given as toxic dosage. And the therapeutic index is of course TD50 divided by ED50. So ideally, a drug should be providing a positive desired effect and at the same time, it should be preventing any adverse toxic effects. So that puts a limitation on the drug dosage and it also provides us with an effective dosage range for any of the drug. So what is the range should be? So look at the figure here. The range is the one which is colored in green. So this is not going into the toxic region and at the same point, it's slightly above the effective dosage. Here is one more concept to keep in mind, the drug bioavailability. Whenever the drug is administered, a fraction, small amount of it will be absorbed in the bloodstream. The percentage of the drug that gets absorbed, that's what we call as the drug bioavailability. Now, which are the factors on which bioavailability depends? One of the main factor is modes of administration of the drug. For example, if you have intravenous versus oral injection, which one will show more bioavailability? Is going to be, of course, intravenous. The next factor is all the medicines, they are chemicals and chemicals may decompose. They also may get metabolized due to enzymes and that can result into less bioavailability. There's also another important factor. The drugs are chemicals and they will have different level of solubility. So the bioavailability also depends upon solubility, and solubility depends upon the functional groups and also the polarity of the compounds. Then there is one more thing. We can always convert the drugs into salt forms and we can make them soluble. Alright, let's discuss each of this factor in detail now. Look at this graph. The graph over here is IV and the one with the lower hump is the oral one. On y-axis we got plasma or blood concentration and on x-axis we got time. So look at this. Within hardly any time, IV gives maximum absorption in blood. Whereas for oral, it takes much longer time and the absorption is much lower than that of IV. So obviously, the bioavailability because of the intravenous administration is 100%, whereas the oral administration gives less than 50% of bioavailability. Other things to keep in mind is about the decomposition and metabolism rate. Many medicines may not be stable at different pH, and as you know, stomach has acidic pH, so some medicines may decompose there. There are also enzymes secreted along the digestive path, and some medicines may react with the enzymes and get metabolized. The solubility of drugs is something which we can control because the functional groups decide upon the polarity of the compound and polarity decides upon the solubility. Just to remind you, we got like dissolves like. Higher the polarity, more is the solubility. So which are those groups which result into higher polarity? Those are the hydroxyl groups, OH, or carbonyl group, or it could be amino group. All these groups will be responsible to add on to the polarity of the compound. Okay, now this one is a magical solution to make the drugs more soluble. What do we need to do? We need to convert them into the salt forms. So there's an example of soluble aspirin. This is the structure of the aspirin which got a carboxylic acid group. And when that is treated with sodium hydroxide, we result into a sodium salt. So here is what happens. We have a carboxylic acid group. And when we treat that with sodium hydroxide, this bond breaks and we end up having a carboxylate group with negative charge, and of course there is a hydrogen ion with positive charge. When carboxylic acid comes in contact with sodium ion, we get sodium carboxylate, and that is a sodium salt which is highly soluble, and that turns aspirin into a soluble salt. Here is one more example of converting a drug into salt form. This is fluoxetine, or more commonly it's known as Prozac. Look at the group here. This is amino group, and as you see, this is a secondary amine. This is treated with hydrochloric acid, and when we do that, the amino group gets protonated. We got positive charge there, and then there is a chloride ion generated that goes and we get a chloride salt. Here is what happens. We got secondary amino group here, and then that reacts with hydrochloric acid. Hydrochloric acid splits into hydrogen and chloride. The hydrogen moves to the amino group and we get protonation of the amino group, which gives you slightly positive charge, and then the chloride ions clusters around that and we get a chloride salt of that amine. So this is another method of making the medicines soluble. So I hope you guys enjoyed the video. I'll see you again in next video. Until then, bye bye.